The overall goal of this procedure is to determine how either a xenobiotic treatment or a disease state alters cellular metabolism.
This method has the potential to reveal how a disease state or an applied toxin affects cellular metabolism. And once this is known, our method has the ability to reveal whether some kind of intervention can correct the defect. The main advantage of this technique is it doesn't rely on transformed cell lines, so it's more likely to provide answers that are directly relevant to human health.
To begin this procedure, prepare a 10 millimolar stock solution of rotenone in dimethyl sulfoxide. Perform a serial dilution of the 10 millimolar rotenone stock solution to obtain solutions with rotenone concentrations ranging from 1 nanomolar to 100 micromolar. Next, add 50 microliters of each stock solution to 5 milliliters of a previously prepared enriched Tyrode's buffer solution to prepare solutions with rotenone concentrations ranging from 10 picomolar to 1 micromolar. Add 50 microliters of DMSO to one of the buffer solution samples to serve as a vehicle control. To isolate platelets from whole blood, centrifuge whole blood samples at 175 times G for 15 minutes with no breaks. When finished, transfer 1 milliliter of the upper platelet-rich plasma layer to a 1.5 milliliter microcentrifuge tube.